Okay, good morning, everybody. We are going to start our chapter on metabolism, metabolics today. So we covered the digestive system last week, and there were homework assignments due over the weekend. And hopefully you guys were able to do those. And what I'm going to do is cover metabolism, the chapter on metabolism between today and Friday. So I've actually moved off the homework, the quiz for metabolism till after Friday's class. I'm going to move the homework as well, um, so that you have, a few extra days to do it. If you've done it, it's not going to affect you at all. Um, but if you haven't done it, I can move that off a few days. Then once we get this done on Friday, I will post the exam on the digestive system and metabolism for you. The other thing that I'm going to do today is post an assignment in Pearson for a lab for digestion and metabolism. So I'm going to look up and try to find some kind of lab that you guys can do, since there's not really anything in your lab kit that correlates with this. Um, and I'll make sure to send out an email when I do that, so that everybody is aware that that's there, because I'm adding that on kind of at the last minute. So without any further discussion, let's go straight to sharing my screen so that we can go straight to the chapter on metabolism. Like I said, I think we'll get through a little over half of this today. We'll have to finish this up on Friday morning. So here we are, chapter 25, metabolism, nutrition, and energetics. So the basic reason that we studied the digestive system, chapter 24, is that we have to talk about nutrients, right? We have to talk about the digestive system is how we get nutrients into our body. So we eat certain foods and make sure that these nutrients get into our bloodstream. And chapter 25 is closely related because it talks about metabolism, nutrition, and energetics. So how do we break down what we eat? What nutrition do we get from it? And how do we use that for energy? Right? That's the main purpose of this chapter. So it just starts off with a few definitions. Nutrients are any essential elements and molecules. Right. So it's just kind of a broad term for things that we need for our bodies to function normally. Metabolic activity or metabolism is when organic molecules are broken down to obtain energy. And the energy is stored as ATP. A big part of why we eat the foods that we eat is to obtain things like carbohydrates. What we do when we get carbohydrates, which are big long chains, is we break them apart into little tiny subunits. And in doing so, we release energy, and we can use that energy for other things in our body. That's a big part of what we're going to talk about today. Okay, and then the energetics part, the last part of this chapter, is more about how the body balances heat gains and losses. So again, it's the energy that's obtained from breaking things down, and then what do we use that energy for? And we'll see that heat is a big part of energy. So to carry out reactions, all cells require certain nutrients, right? They require oxygen, water, vitamins, minerals, and we get these from the foods that we eat. And if I asked anybody, what's your metabolism, or how would you define metabolism? It's, usually most people think of, well, it's how well I break things down, how well I break down what I eat. But it's actually the combination of these two terms, catabolism and anabolism. Catabolism is what you're thinking of. That's how you break things down, right? Anabolism is then taking those individual pieces, those individual subunits, and building them back up to materials that you need. So think about when we eat something, we eat these big long chains. We break them apart into their individual links, and then we put those links together in a different order for the materials that we need. So breaking that chain into the individual links is catabolism, and then putting those links back together in a different order is called anabolism. The combination of both of those is your metabolism. So it's how well you break things down and then use those products, right? And put them back together in a different order, rearrange those products in a different order for the molecules that you need in your body. Right? So here are basically our nutrient pools. Everything we eat is either a fat, a carbohydrate, or a protein. And you can see how they kind of feed into different things, right? Obviously, carbohydrates feed into this glucose pool, which is used for energy. So brain metabolism and metabolism in most tissues. Fats as well can be used as metabolism in most tissues. Proteins, which are amino acids, are much harder to get to this pool of metabolism. So this is only when we're talking about ATP energy. It's carbohydrates are the easiest, which we'll see. That's what we kind of talk about first. Fats are the second easiest, right? If there's no carbohydrates around, you can use fats. If you don't have carbohydrates or fats, your body will use proteins. Proteins are the hardest to convert into energy. So again, metabolism is converting large molecules into small ones. You take a big chain, break it down into individual subunits, and when you do, you release energy. And anabolism is taking those individual subunits and sticking them together to make a chain that you need. So you stick them together in a different arrangement or a different order than what they were broken down from to make molecules that you need. That's considered uphill, right? You need energy to do that. So catabolism releases energy. When you break down those long chains, you release energy. And then you use that energy to link things together in the formation that you need. And so, why do we have anabolism? Why do we need to build things up? Well, we need to maintain and repair a lot of our tissues, right? Think about if you scrape your skin, you need to grow new skin cells there, that sort of thing. This also supports growth. So you do a lot of anabolism as a child, as a young person, an infant, a toddler, a child. You have to grow, your bones have to grow, your muscles have to grow, all those sorts of things. Produce secretions, so things like hormones, other types of secretions like enzyme secretions, those are produced by anabolism. And then storing nutrient reserves. We'll see that we actually store some of the excess nutrients that we have, we link them together and we store them somewhere for later, basically. So what do we have for nutrient reserves? Anytime we have too many, too much of a certain nutrient, we can store that and use it later. And there's three main ones that they talk about here. For lipids, right, our energy reserves, our nutrient reserves are triglycerides. These are the most abundant storage lipids. They're mostly made of fatty acids. We'll see later on in this chapter that they can actually be converted into energy. We also have nutrient reserves that include glycogen. Glycogen is a carbohydrate storage. So when we eat a meal, we have a lot of glucose in our blood. We can link those glucose molecules together as this molecule called glycogen. We can store glycogen in our liver and in our muscles. And then we have proteins, right? Proteins are very abundant. All of our enzymes are made of proteins. They perform a lot of vital functions in the body. So when we're talking about what we're using, triglycerides are the main type of lipids that we're talking about. Glycogen is the main type of carbohydrate, and proteins are the main, enzymes are really the main type of proteins that we're talking about. Energetics is the study of the flow of energy, as the name suggests, from one form to another. And in that, we have to talk about these reactions called oxidation reduction reactions. You have reactions in your body that take place where there's the oxidation of one of the starting materials and the reduction of another. They always occur simultaneously. You don't have just oxidation or just reduction. If you oxidize one starting material, you are reducing another. Okay. They are always paired. So a lot of times they're called redox reactions just because it's easier to say than oxidation reduction. Basically what happens is you are moving electrons, and electrons carry chemical energy. So you start off with Here's an example of this. If you start off with one reactants, right? You have two reactants on the starting side of the equation. One of them is going to lose electrons and the other is going to gain electrons. So that's essentially that's what you're doing. You're moving electrons from one substrate to the other. The one that is oxidized is the one that loses the electrons. The one that's reduced is the one that gains the electrons. It seems counterintuitive to say that the molecule that is reduced gains electrons. But here's the thing. Electrons have a negative charge, right? You should have learned that way back in chemistry, that electrons carry a negative charge. So if you are a molecule and you're gaining electrons, you're gaining negative charges, right? So that's said to be reduced. So any molecule or atom that gains these electrons is gaining energy. The electrons are energy. They're carrying energy. But that molecule is said to be reduced, right? If you are losing electrons, it's said to be oxidized. And then there's always some energy released as heat. So no system is completely 100% efficient, right? Car is not efficient. Nothing is 100% efficient. So in converting this energy, right, and moving these electrons from one molecule to the other, some of that energy is always lost. Not 100% of it does not get transferred. And the part that's lost is usually released as heat. We will also talk about the electron transport chain. The electron transport chain is something that occurs in mitochondria. So mitochondria are little organelles within a cell, right? They're the organelles that carry out energy production. And part of what they do is they take these electrons and they pass them through a series of redox reactions, right? They hand them down this chain from one molecule to another. And what happens is as these electrons are being transferred from one molecule to another, they're releasing energy. And the mitochondria are taking that, right? They're taking that energy that gets released and they're storing it as a molecule called ATP, which is going to become really important. That's the molecule that we all use for energy for all of our cells. The electrons eventually end up binding to oxygen, right? And that forms water. Think of electrons, always think of electrons as hydrogen. And so if you take hydrogen and oxygen, that makes water. So the end of this, right? You take these molecules that have electrons and you pass them through these different atoms. And at the end, they all bind to oxygen to make water. But in that process of going through all those steps, you're releasing energy, you're releasing ATP, or you're releasing energy, which is stored as ATP. There are coenzymes that play a key role in the flow of energy within a cell. Coenzymes act as intermediaries. And so what happens is you take these electrons from one molecule and you stick them onto a coenzyme. Now the coenzymes that we'll be talking about are NAD and FAD. And so what you are actually doing, think of electrons as hydrogen molecules. Think of them as H's. So you're taking H's from something like glucose, and you're sticking them onto NAD to make NADH, or you're sticking them onto FAD to make FADH. The NAD and the FAD are just holding on to those electrons, bringing those to the electron transport chain so that you can produce ATP from them. They're just intermediaries. They're just holding on to those electrons. Ultimately, those electrons are going from glucose to oxygen. Remember, oxygen is the final acceptor, right? So you're passing those electrons from glucose to oxygen. In the middle, they're getting passed through either NAD or FAD, which will help you to make ATP from them, will help you to make energy from them. Right? FAD can become FADH2, so it accepts two hydrogens, right, H2, and those two hydrogens represent two electrons. always think of electrons as hydrogens. They're always hydrogens in this context. Coenzyme NAD can accept one, so it forms NADH. So here we get to carbohydrate metabolism. This is where all of your cells contain mitochondria, and what the mitochondria do is they try to take glucose and oxygen and they make energy from it, right? They break it down to release energy from it. So carbohydrate catabolism, right? Carbohydrates are long chains and you're breaking them down into individual units to release energy. What this does is you can generate ATP, right? You can take that energy and store it as a molecule called ATP or other high energy compounds, but mostly what we're going to talk about is ATP. And this is the equation that all cells use, right? That all cells want to use, prefer to use, I should say. Glucose plus oxygen goes to carbon dioxide plus water. Right? When you take H's from glucose and you stick them on oxygen, what you're left with in glucose is just carbon and oxygen, which is carbon dioxide. When you stick those H's onto oxygen, they become water. Right? So that's how glucose and oxygen go to carbon dioxide and water. Now, glucose is giving up electrons, right? It's going to give those electrons to oxygen. So glucose is actually being oxidized. Oxygen is gaining the electrons. Oxygen is being reduced. Right? This process involves 3 steps. And those 3 steps are glycolysis, the citric acid cycle, and the electron transport chain. So this takes place inside the cell. The cell is going to take glucose and oxygen and be able to break apart that glucose, break it down, right? Glucose is 6 carbons stuck together. And what's holding those carbons together is energy. And so if you can break those 6 carbons apart so that you have 6 individual carbon molecules, you're releasing energy from in between each of those carbons. And that's what you're trying to do with cellular respiration. So to get to that point, the cell undergoes these 3 steps, or these 3 stages: glycolysis, the citric acid cycle, and the electron transport chain. And the end of that, what you make is ATP. So you start off with glucose and oxygen, and you release energy, which makes ATP. It gets stored as ATP. Now, for every 1 glucose molecule, you can make somewhere between 30 and 32 molecules of ATP. That's pretty good. As long as you have oxygen around, you can do that. Now, these other 2 things are just byproducts, carbon dioxide and water. We have to get rid of them. Well, carbon dioxide builds up in our cells and we exhale it. That's how we get it out of our body. And water, right? Water gets passed to our urine. We urinate it. That's how we get water out of our body. So in this first step of carbohydrate metabolism, the first step is called glycolysis. Glycolysis just takes glucose and it starts to break it down. So glucose is a molecule that's got 6 carbons linked together, right? And some oxygens and hydrogens. What you want to do ultimately is break that 6 carbon molecule into individual carbons. The first stage, glycolysis, just takes that 6 carbon glucose and splits it in half. So you have 2 3 carbon molecules. Right? That's all it does. It breaks the 6 carbon glucose down into 2 molecules, and each of those are 3 carbon molecules. And they're called pyruvates, right? Or pyruvic acid, same thing. This first step does not require oxygen. Remember, you have glucose plus oxygen. You only need the oxygen for the citric acid cycle and the electron transport chain. Glycolysis, this first step, happens even without oxygen. So it's in the cell that this is going on. It's not in the mitochondria. It's just in the cellular, in the cytoplasm, inside the cell. But you're taking a 6 carbon molecule and you're breaking it into 2 3 carbon molecules. right? And there's an enzyme that helps this process. And so what do you need for glucose? You need glucose, or what do you need for glycolysis? You need glucose, you need certain enzymes, you need energy, ATP and ADP, you need inorganic phosphate groups, you need this coenzyme NAD, right? Here's what's happening. This is your glucose molecule up here, right? In purple, 6 carbons. All I want you to know is what happens, what you start with and what you end with, right? What you end with are these 2 glyceraldehyde 3-phosphate molecules. How you get there, right? You put a phosphate on here, which is glucose 6-phosphate. You put another phosphate on the other end, right? And then it gets split. These middle steps, I'm not going to test you on those. But what you need to know is that glycolysis takes this 1 6-carbon glucose and you end up with these 2 3-carbon molecules. Okay. So it goes through all this. I'm sorry, this isn't the ultimate end. The ultimate end is what happens here, where you get these pyruvates. So there's a lot of addition and subtraction of phosphates onto different molecules, right? And eventually you end up with these 2 3-carbon molecules called pyruvate. Now, here's the energy summary. This is within glycolysis. The first 2 steps of glycolysis cost 2 ATP, right? In order to get through step 1 and step 2, you have to put in 2 molecules of ATP, 2 molecules of energy. When you get to step 5, however, you get 2 molecules of ATP back, right? The step 5 creates 2 molecules of ATP. Step 7 creates another 2 molecules of ATP. What that means is that the net gain is 2 ATP. If we're talking about what you start beginning here, right, and what you end with down here, to get from the beginning to the end, you end up with 2 more molecules of ATP at the end than what you started with at the beginning. That's what I want you to know. Okay, you make 4, but you spend 2 of them, right? And that's what this is showing you in the summary. So the net gain for this first stage of catabolism, of carbohydrate breakdown, is 2 ATP. Okay, the first stage is called glycolysis. It doesn't use oxygen, right? It doesn't take place in the mitochondria. It's just in the cell. And what you end up with are 2 ATP. Now, you also, look, there's 2 NAD here that get transformed to 2 NADH. So in addition to making 2 ATP, you make 2 NADH. Now these are coenzymes. These are going to get saved for the 3rd stage of glycolysis, or I'm sorry, for the 3rd stage of cellular respiration. Right, so we are only talking about the first stage. We're only talking about glycolysis. During glycolysis, you make 2 ATP and you make 2 NADH. Those 2 NADH get saved and get used in the electron transport chain, that 3rd stage. Okay. So if we're talking about what glycolysis does, it makes 2 ATP and makes 2 NADH. Now, then we get into aerobic metabolism. We get into what happens when you have oxygen around, right? Aerobic means you have oxygen around. Just like, aerobics, the 80s exercise craze, right? So this occurs within the mitochondria. Whenever you're talking about having oxygen around and aerobic respiration, aerobic metabolism, this happens inside the mitochondria. So the first stage, glycolysis, didn't happen inside the mitochondria. It was just in the cytosol of the cell. The second stage, and we'll see the 3rd stage, happened in the mitochondria. This does require oxygen. And what you're going to now do is take pyruvate and break it down even further to make more ATP and more of those you're going to pass more electrons on to NAD and FAD to make them NADH and FADH. Right, and so there is an outer membrane and an inner membrane. So the outer membrane is permeable to ions and small organic molecules. The inner membrane contains the carrier proteins. The inner membrane is what's going to be really useful in the third stage of cellular respiration. So in the citric acid cycle, you take your hydrogen atoms of pyruvate and you remove them by coenzymes, right? So you produce a lot of that NADH and FADH2. The carbons and the oxygens are removed and released as carbon dioxide. And what happens in the mitochondria, pyruvate interacts with NAD and coenzyme A, and you get acetyl-CoA. Now, that acetyl-CoA then goes into the citric acid cycle, right? One citric acid cycle removes 2 carbon atoms, regenerating a 4-carbon chain. Several steps involve more than one reaction or one enzyme. Here's the whole thing. Now, you've got a pyruvate. It gets converted to acetyl-CoA. It goes through the citric acid cycle. Now look, as you're going through the citric acid cycle, you make NADH, you put an H on to make NADH, you put another H here to make NADH, you produce an ATP, right? And then you have 2 more H's to make FADH2 and another NADH, right? So in addition to making 1 going from pyruvate to acetyl-CoA, you made another 1, 2, 3 of NADH. So now you've got 4 NADH, 1 FADH2, and 1 ATP. Now that's only for 1 pyruvate. Remember, glucose produces 2 pyruvates, right? You split that glucose into 2 pyruvates. So you go through this citric acid cycle twice for every glucose. So, and again, I don't want you to know all of these intermediate enzymes in this class, but the end result, what you need to realize is that all of these NADH molecules and the FADH2 molecules, those all go to the electron transport chain, right? So remember, you made 2 NADH in glycolysis. Now you're making another 8 NADH, right? Because 4 here plus 4 for the other acetyl-CoA, plus FADH2, you're making 2 molecules of FADH2, right? Because you're going through this cycle twice. All of those NADH and FADH2 molecules get transferred to the electron transport chain. So far, we've gone through the first 2 stages. We've gone through glycolysis and citric acid cycle. We made 2 ATPs in the citric acid cycle and 2 in the glycolysis. So we've only made 4 ATPs so far, but we've produced a lot of NADH and FADH2, which is all getting shuttled to the third stage. The third stage is where we are going to basically cash in on all of those coenzyme molecules to make more ATP. The third stage is called oxidative phosphorylation. This is where we can actually generate ATP from NADH and from FADH2. Okay. It occurs in the mitochondria. You have to have oxygen for this to occur. This produces over 90% of ATP used by the body. Remember, at this point, we've only produced 4 molecules of ATP. That's not that much. But during this, we're going to produce like another 28, 30. This is the basis for the formation of water. Remember, you need oxygen, right? I keep saying you need oxygen. And here's the oxygen right here. You are always going to think of electrons as hydrogens. So these electrons that are stuck onto NADH, they're these H's. And the ones that are stuck onto FADH2 are these H's. Those H's are going to get transferred and bind to this oxygen to make water, right? That's where the water comes from that we have to get rid of. And what I'm going to do is go to this picture. A better way to do this. Okay. So we're going to go to this picture right here. This picture is showing the mitochondria. In tan up here is the cytosol. It's outside the mitochondria. Here's the outer membrane between the mitochondria and the cytosol. And then remember, the mitochondria has a second inner membrane, right? So here's the space between the two membranes. And in the inner membrane, there are a bunch of molecules that are embedded in that membrane, okay? Those are carrier molecules. Carrier molecules can push things through a membrane, push things across a membrane. And so what happens is the electrons from NADH, which are the H's, those get pushed through this carrier, right? So they get, NADH is in here in the inside of the mitochondria, the inner membrane. Those H's get pushed through that inner membrane to get pumped into the intermembrane space. The same thing happens with the H's from the FADH2, right? All these H's are getting pumped into this intermembrane space. So this intermembrane space is filling up with H's. The H's are being pumped from the mitochondrial matrix, the inner part of the mitochondria, into this space between the two membranes. So the space between the two membranes is filling up with H's. Now, what direction do they want to move? Well, molecules always want to move from where there's a lot to where there's very few. So what you're creating is a situation where there are a lot of H's here and very few in here. And so those H's are going to want to move from the intramembrane space to the mitochondrial matrix. Now you have these pumps, keep pumping them that way. The only way for them to flow back into the mitochondrial matrix is through this last carrier here. Right? This purple molecule all the way on the right is called ATP synthase. And it has a proton channel. The H can actually fit through this channel. And when it does, every time an H goes through there, it's like a turbine that turns, right? And as it turns, it converts an ADP and a phosphate into an ATP. So for every hydrogen that moves through, every hydrogen that moves from this intramembrane space into the mitochondrial matrix, you're producing one ATP. So when you think about all the hydrogens that you made for NADH and FADH2, for each one of those, you're producing an ATP. And so in doing this, you are producing the majority of your ATP. Remember, in stage 1, you've only produced 2. In stage 2, you've only produced another 2. In stage 3, you produce something like 28 molecules of ATP or glucose. Okay. So the hydrogen ions, they get pumped into that intermembrane space, right? Those are the molecules that are embedded in the membrane are constantly pumping those hydrogen ions into the intermembrane space. And what happens is those hydrogen ions then want to diffuse back into the matrix. And so they do. And when they do, every time they pass through, every time one hydrogen passes through, it produces an ATP. And so the NADH molecule, right, it pumps 6 hydrogen ions into the intramembrane space. The re-entry into the matrix generates 2.5 molecules of ATP. These numbers are hard to estimate because there's so many hydrogens moving, right? The FAD, right, this was the molecule that became FADH, that pumps 4 hydrogens into the intramembrane space, and re-entry generates 1.5 molecules of ATP. So this oxidative phosphorylation, where we cash in on the NADH and the FADH2, that's really where we're producing most of the ATP. It does require oxygen and it requires electrons, right? If you are low in oxygen or low in electrons, you're not going to be able to generate as much ATP. But the energy yield of glycolysis and aerobic metabolism, for most cells, this is the main method of generating ATP. Okay, and so it starts with glucose and ends with carbon dioxide and water. Here's glycolysis, right? One glucose molecule gives you 2 ATP and 2 NADH. Citric acid, you have 2 revolutions of the citric acid cycle because you're producing 2 pyruvates. So each revolution gives you 1 ATP. So there's an additional gain of 2 ATPs because there's 2 revolutions. You transfer H's to NADH and FADH2. Right. Then finally, the electron transport chain. It tells you what you produce from the NADH and the FADH. Total yield is about 23 ATP. So the summary for each glucose molecule processed, the cell gains 30 to 32 molecules of ATP, right? 2 from glycolysis, and then you generate NADH in glycolysis. 2 from the citric acid cycle, and 23 molecules of ATP from the electron transport chain, from that third stage of cellular respiration. All but 2 ATP are produced in the mitochondria, right? The first 2, the 2 from glycolysis, are not produced in the mitochondria. They are in the cell outside the mitochondria, in the cytoplasm. And so this is just a summary of that. So what I'm going to do is I'm going to leave it there because that's a lot to go through for one day. I'm gonna stop there. Stop here. I'm gonna stop there. We're gonna leave it there. We're gonna meet up on Friday to go through some of the rest of that chapter on metabolism. But study up on the three stages of cellular respiration that we went through. And what I'll do is I'll move the due date for the homework assignment. I'll also create a lab assignment in Pearson, and I'll email everybody so that everybody's aware that that's out there. And if you have any questions that you think of, just please email me, let me know. And if I don't hear from you soon, I will see you on Friday. Thanks.